So what I'd like to do today is thank you very much, Peter, for the introduction. And I was counting. I had a little pre-speaker intelligence that he would cover RSV, and so he's given some very nice background. I thought I would touch on the background at a meeting like this with the time limitation. I can't go into all the details of our phase 2 data, but we recently announced some phase 2 data, and I will give you sort of a top-line summary, frame it as to what I think is important, talk a little bit about Dr. Sieber's work. I was a really nice surprise to see him chairing this meeting. Talk a little bit about the cotton rats, and then our clinical development plan, where we're going. So, from my standpoint, I used to be useful. At one time in my life, I was a pediatrician and saw patients. I saw a lot of RSV. I used to moonlight downtown D.C., and RSV is a predictable epidemic in the wintertime, and it really does affect children, certainly up into school age. You could see the sequelae of wheezing afterwards. And the way we've divided the target populations into RSV from a vaccine standpoint is we think we can address infants, and I'll talk to you about the hospitalization rates through 0 to 5 months via maternal immunization, where we'd immunize women in their 3rd trimester and protect the infants via transplacental transfer. The other population, which I won't address today, is infants and children, be 5 months to 5 years, where I could see an annual seasonal vaccine. And as you may know, we have also a ongoing clinical trial in elderly and high-risk adults, which I won't touch on today. So RSV is a very common viral infection in winter, causing bronchiolitis, pneumonia. What's the hallmark, as you see, as I'm again at a disadvantage, so you don't have a pointer, but it's there, plugging of the small airways with necrotic debris and virus. The sequelae are wheezing, apnea, otitis media. And again, Peter touched on this. It is a global disease. We're concerned. We have a partnership with PATH, and I think, in the U.S., the burden of disease is very high. And in my scratching of the surface of the epidemiology, both through travel and discussions with other people, I think RSV is even more, has a higher incidence in many settings throughout the world. In the U.S., some of my colleagues have taken a look at the National Hospital Discharge Survey, and I think this points out a very important set of epidemiology for the talk today. You can see on the left the U.S. hospitalization rates, and then the age in years, and then on the right we've amplified that down to age in months. And you can see RSV is a very intense disease in the first few months of life. In fact, by 6 months of age, 80% of all hospitalizations from RSV have occurred. So this makes a challenge because this is a population that's hard to immunize. And so, coming along with this challenge, 50 years of RSV vaccine development, tremendous amount of work has been done. As Peter mentioned, there was the unfortunate experience in the 60s with the formalin-activated vaccine. On top of this, you have these very high hospitalization rates in the young, where they're fragile, their airways are small. And then on RSV, as Peter mentioned, you see a lot of recurrent infection, and the RSV itself has strain changes from year to year, making this a very taxing and complex problem, pathophysiology, and from a vaccine standpoint, really a challenge. I think maternal immunization addresses many of these challenges for at least the population under 6 months of age. They have small airways. They have a time at which they are protected from the mother's experience with natural infections. So they do have antibodies, but there's a bell curve. Once those antibodies wane, they become very susceptible. They're very challenging to immunize. I think they have a relatively poor immune response. We need to have rapid and full immunity to address this, and we know that, as I mentioned, maternal antibodies can be protective. I think one of the things that over the years that I've been looking at this problem is that the monoclonal antibody Synagis or palivizumab are known to be protective, and that's a very important lessened. So there we have passive immunity given to preterm and term infants, which are protective, and I think there are important lessons for us in vaccines to derive from this. A few comments on maternal immunization. So antibodies are actively transferred from the mother's blood to the baby's blood through the syncytial trophoblast. The concentration itself begins about 28 weeks gestation, so it's really active in the last term of infants. And most infants at birth, again, depending on what you measure and how you measure it, have well over the levels you find in the mother's antibodies. And in fact, the concentration effect can be quite dramatic, especially with protein antibodies. We know from tetanus, for example, that they can have up to 180% of the mother's antibody levels. So that's good, and that helps us as we move forward. And we know, of course, in the context of RSV, that there is maternal protection through maternal transfer of antibodies. So, current practices, again, this is not a new topic. It's been around for a while. It was employed very effectively by the WHO for a maternal neonatal tetanus campaign, where they have eliminated neonatal tetanus to the tune of 92%. So as you might imagine, as a baby is born in a very poor setting, the umbilical cord tied off with a string, they become susceptible to tetanus, and this has been a very effective program. Influenza, as was earlier mentioned, is now given universally, and then pertussis was recommended to be given in the third trimester to all women to protect the infant against pertussis. So as we've pondered a clinical strategy in a very complex field, you can see we've moved along. We've done a healthy elderly trial, phase 1 trial, and we published that in December. And the trial I'm going to report on today is a women of childbearing age trial, and what we're moving towards is the third trimester immunization of women to show efficacy in infants. So my little cartoon of the RSV budding off, you can see there's two major surface glycoproteins, the F protein and the G protein. The F protein is involved in viral entry and syncytia formation. The G protein is attachment. The F protein is very conserved across strains. In particular, the epitopes that palivizumab target are highly conserved across strains from year to year. And as opposed to the G protein, which has a great deal of variation from year to year, different subgroups, and makes maybe one of the explanations for the problem of recurrent infection, which we all have had infections, yet we still continue to get RSV disease. So a few comments about palivizumab. Palivizumab is a monoclonal antibody. It binds to a site on the F protein and the F trimer, and it's been shown to prevent RSV disease in multiple studies, 2 randomized clinical trials. I would also add that modivizumab, which is targets the same epitope with higher affinity, has also been shown to be protective against RSV disease. And one of the interesting findings from our vaccine trials, and I would say this is an original piece of knowledge, but I think we've been able to highlight its importance, is when people have natural infections, so you would have had 30 years plus, depending on how old you are, of RSV infections, yet when we look in the general population, we do not find palivizumab antibodies in that population. So somehow it's cryptic to the immune system during infection, in fact, from repeat infection, and I'll show you that in just a second. So at Novavax, we make different particles, virus-like particles, which are what you would imagine. They look like a virus. For example, the flu, which has the M protein and HA and NA spikes. And then the protein nanoparticles, which really are just a single antigen in a repeating unit. They're engineered so that they coalesce into a particle. They have repeating subunits with the epitopes in their native configuration. They make a highly immunogenic particle and should induce antibodies that are functional. So I'm going to talk a little bit about our phase 2 data in women of childbearing age. Just again to remind you, our target product profile is protection of infants against RSV disease in the first few months of life via maternal immunization. So this is a very important jump for us to go to this target population. So we had study objectives. First of all, safety, which is important to enlarge our safety experience. And then secondly, we had a number of vaccinology questions that we needed to answer. Is there an advantage of aluminum phosphate adjuvant, which we'd used in the first trial, 2 versus 1 dose and increasing the dose. So we evaluated that in the context of anti-F. It has a very high dynamic range. In our view, in our assay, we think it's a functional immune response. We also looked at microneuts, and then we also have developed the palivizumab evaluation through palivizumab competitive ELISA and other assays. Fairly complex study. It was matrix, so we could have power to answer a lot of questions. But essentially, we did 60 and 90 micrograms, either with alum or without alum, and they were either dosed twice or once, and the second doses were always placebo. The dark brown bar is the phase 1 formulation. One of the things we were able to do in this trial is advance the formulation to a pre-manufactured, pre-absorbed formulation that was then shipped to the site. The phase 1 trial was done as a bedside mix, and you can see we have placebo-controlled. I apologize for not going in detail for the safety. It's just time limitation, but the demography, we had all females. The average age was 26. They were stratified, so we had half 26 to 35, and the other half were younger. The safety results were really confirmative of the phase 1 data, and I just showed the publication headline down below so you can look it up, and we intend to publish it. Really, what you would expect from a recombinant vaccine: mild injection site pain, no SAEs. We had one fever 17 days out of vaccination. So really, I think quite a quiet vaccine from a safety standpoint. Now, looking at the primary immunogenicity endpoints, I've divided them up into the 1 and 2 dose regimens because it's quite interesting. First of all, the very bottom line are placebos, and you can see the anti-F IgG response from years of natural infection, mean age of 26. So they tell me, Peter, they probably had 20 RSV infections over their lifespan, and yet have very low anti-F antibodies by our measure. Yet when we vaccinate, even at 7 days, we see a boost, and by day 28, which is in the middle there, you can see the peak antibody response after 1 dose. So the top 2 are the higher doses with adjuvant. The bottom 2 are lower doses without adjuvant, and you can see, as you might expect, the antibody response start to wane. Same kind of graph now here. Different groups, however. You might expect the same sort of picture. Day 7 immunization or bleed has an increase in antibody response. And out at the day 56 after the boost, you can see more differentiation. The 3 bands there are the 2 aluminum phosphate adjuvant groups that are pre-formulated, and the phase 1 is the orange bar, and then down below the non-adjuvant groups. So we got very clear questions. These are really important for us going forward. Aluminum phosphate had a positive effect. It was a highly significant effect, and that was seen at either 1 or 2 dose regimens. When we looked at 2 versus 1 doses, as you might expect, the aluminum phosphate 2-dose regimen was better than 1, conferring about a 1.4-fold rise. And then finally, was there increased immune response at 90 versus 60? It was very, I think, minimal, suggesting we are at the top of the dose range. Now, microneutralization is another assay that's used commonly in RSV-ology, I should say, and you can see here, this is the RSV A long microneuts. Again, this is our primary immunogenicity point, was at a day 28, and these are reverse cumulative distribution curves where you see the percentage in the subject on the x-axis, on the y-axis, and the microneut log 2 titer on the y-axis, and what you can see on the bottom bars, those are the placebos. The middle bar are the vaccines without adjuvant, and the adjuvant groups are pooled together. You can see in the far right, very nice adjuvant effects. This is a very noisy assay. With the B, it's not so evident at the day 28, but again, we see movement. What we're trying to do, again, I wish I had a pointer, the microneut titers show a reduced risk of hospitalization where they're log 2.6 or greater, so we're able to get rid of all the low titer subjects by vaccinating as by this measure. Turning to palivizumab, the palivizumab is a competitive ELISA assay where we can then convert our findings to a standardized curve, which I show in the bottom right there. So every ELISA unit is about 2 micrograms per ml of palivizumab equivalent. And you can see there, first of all, I would point out the day 0, those 35s, those are at the limit of quantitation. And when we look, if we just lift that barrier, you can see there's almost no response to palivizumab at the day 0 and the placebos from the general population. So this is 300 plus subjects, many years, multiple years of repeat infection with RSV and no palivizumab antibodies measurable. By contrast, after immunization, and I circled 3 that were the adjuvant 2-dose regimens. This is very similar to what we saw in the phase 1 trial, reaching 300s microgram per ml. And I'll talk about what might be a protective level. So now we begin to get into the exploratory endpoints and think a little bit about what this might mean. As I mentioned, our target product profile is to immunize mothers. The antibodies would be transplacenally transferred, and then there'd be some decay. So when you look at the literature, there's a variety of ways to look at this, the half-life, the concentration. So we've made some assumptions here, and just taking our best case, which I can do today, we can see at the 90 microgram dose, the lower bound of the confidence interval of our antibody titer, our concentration, or palivizumab-like antibodies, assuming 30 microgram per ml is protective, takes us out well into 5 months of age, which is really, a very gratifying result. In fact, I gave the best case. It doesn't make a lot of difference on the different vaccine regimens at 1 or 2 doses. It's really around 4 to 5 months where we see these begin to cross. So you can just amplify it there for you to see. And this is important. As I mentioned, 80% of the hospitalizations have occurred by 6 months of age. So we're really in the sweet spot where we want to be with antibodies we think are protective. So, in conclusion, the safety immunogenicity really resembled our phase 1 data. It was well tolerated with just local AEs is the main finding, as we would expect. The anti-F microneutralization antibodies were similar or superior to what we saw in phase 1, and we answered our questions that aluminum phosphate is helpful, 2 doses are superior to 1 when alum is used, and we didn't really need to go up in the dose. So I'd like to just take a few moments to pause and look at some history of the cotton rat, because I think it's very helpful for us. While the cotton rat does not get RSV disease, it does represent, in my view, a very good in vivo assessment of the vaccine, of the potential vaccine efficacy. And I point back here to an old paper here by Dr. George Sieber, sitting in front, where they looked at Respigam. They evaluated Respigam. On the top, you can see this is passive immunity, passive antibody given to cotton rats, challenged with RSV in the lungs, and now you're looking at lung titers, and as you go to the right, you have decreasing concentrations of your antibodies. So you create an S-curve, a dose-response curve, and they use this information to license a product, the first product used in premature infants. This type of study was followed with the work by Johnson et al., again in cotton rats, again describing an S-curve, finding a level that was protective. And this protective level was used heavily by MedImmune as guidance as they moved out into their clinical trials. And I must say, this is also used for Novavax. So we think that the cotton rat is extremely helpful in terms of determining what might be a protective level going forward. When you immunize a cotton rat with the F nanoparticle, get very good F antibodies. You can see here the groups include the formalin activated vaccine, the RSV infection, which is a control. We know infection is protective against homologous infection. Our vaccine with alum, without alum, with placebo, and passive. And the placebo and the FIRSV are right at you can see at the bottom there, the geometric means are at the limit of detection in these assays. So our vaccine induces very high neutralization. In contrast, the formalin activation, I'm not sure that there is the barrier that may have been to use of a protein vaccine in seronegatives, but that's a different day, different topic, and you can see palivizumab giving neutralizing antibodies. When you measure the palivizumab antibodies, you can see from the formalin activated vaccine, there's no measure there, some with the RSV infection, and the vaccine is very high, and the palivizumab is in the range that's protective, but much lower. And then when you look at challenge of the cotton rat. You can see in the groups, as you might expect from our assays, the vaccine is highly protective, as is infection. Formalin activated is not, and Pali is not. When you look at the nasal titers, you can see, as you might expect, the live infection is effective, as is the vaccine, where we do not detect virus at all, and palivizumab, there's partial protection. So this was a very good trial for us. I think the phase 2 reproduced the phase 1 data, which is remarkable, given all the different assays we've developed, the different formulation. It's moved the program into the target population. We've tested an advanced formulation. We expanded the safety database. And as I was trying to allude to at the end there, I think building in this relevant preclinical model and assays, in particular the palivizumab assay, is very helpful for de-risking the program and helping us plan for going forward. And with that, I'll thank my many collaborators at Novavax, and also would like to thank PATH for support of the phase 2 study and the work done by Sigmovir on the cotton rats. With that, I'll take questions. Thank you very much.